Dr. DeBellis, and I'm going to talk about the action potential. In order to do this, first I'm going to tell you about the parts of the neuron. A neuron is a cell that's specialized. It's a cell that's capable of sending a signal. We're going to talk about the qualities that make this possible. So this is a standard neuron. As you can see, the flow of information will be from left to right. A neurotransmitter typically activates a receptor channel that's in the dendrites. This happens at these little dots. We're going to call them dendritic spines. And this allows the charge to change. The depolarization or hyperpolarization of the neuron occurs here. And it moves in a graded manner from the dendrites to the cell body. This is the cell body here. It encases all of the organelles of the cell. It's where the nucleus, endoplasmic reticulum, all the organelles are at. This is the axon, hillock. That's where we first see voltage-gated sodium and potassium channels. This is where the action potential actually begins. So when we talk about the action potential, we're talking about this particular part of the neuron. This is the axon here. Again, information's going to flow from left to right. You see these little beads. These little beads are myelin. They insulate the axon. We have these little tiny exposed areas, what we call the nodes of Ranvier. The electric signal travels from the left to the right, and finally it reaches the axon terminals. And at the terminal buttons, which will be the exit point for the neuron, that's where specific neurons, or the axon terminals are where specific neurotransmitters are released into the synapse. So to remember the different types of pumps and channels that enable the neuronal membrane to conduct information, we have the acronym PSVV. The P stands for potassium channels. These are passive channels with selective permeability to potassium. Potassium can travel both in or out of the neuron through the potassium channels. The sodium-potassium pump, on the other hand, it pushes sodium out of the intracellular space while simultaneously pulling potassium into the intracellular space. The final two channels are voltage-gated. They allow the action potential to occur. First, we have the voltage-gated sodium channel. The voltage-gated sodium channel is permeable to sodium, and it opens when the interior of the neuron reaches -40 millivolts. It closes when the interior of the neuron reaches +40 millivolts. At that time, the charge of -40 millivolts on the interior of the neuron opens the voltage-gated potassium channels, which are selectively permeable to potassium, which is then forced out of the intracellular space. It runs down its diffusion gradient. It runs down its concentration gradient, and the potassium travels to an area of lower concentration. It's also drawn out of the intracellular space by its charge, because at this point, the charge inside of the neuron is too positive. And that drives the positive potassium cations out of the intercellular space. Potential refers to the fact that the neuron has a negative interior. You can probably tell by this diagram of the grumpy neuron that not only are neurons excitable or irritable, they're also very negative. The charge of a neuron is typically negative 40 or negative 65 millivolts. When the action potential is not occurring, we say that the axon of the neuron is at a resting state. At the resting state, the exterior of the neuron and the interior have different concentrations of cations and anions. A cation is a positively charged ion. That's why I have the plus sign, the positive sign. And anion is negatively charged. As you know, positive and negative charges are attracted to each other. Like charges are repelled. So cations push away other cations, and anions push away other anions. This is one of two major forces that's at play when we talk about the action potential. The first is electrostatic pressure. The other is going to be diffusion. That's the fact that substances travel from an area of high concentration to an area of low concentration. This is a dam, for instance. In the case of the dam, the water before the dam is in an area of high concentration. If you remove the dam, it goes to an area of low concentration. The water is said to be traveling down its concentration gradient. When at rest, the axon has a negative charge. The major reason is that it has proteins. And these proteins are negatively charged. There's also a considerable amount of potassium. And potassium is a cation. And we're going to draw potassium in here. There's a little bit of a potassium outside. However, we tend to see more sodium on the outside. That's another cation. Potassium channels have what we call selective permeability. That means that potassium can travel either direction. Initially, potassium travels to the interior of the neuron because potassium is positively charged and the interior of the neuron is negatively charged. Therefore, electrostatic pressure pulls it to the interior of the cell. But at some point, there's so much potassium that it begins to leave the inside of the neuron. It travels down its concentration gradient. There's so much potassium here on the inside of the neuron that it travels down its concentration gradient to the exterior. Now remember, we also have the sodium-potassium pump. And what the sodium-potassium pump does is the sodium-potassium pump pulls more potassium in. It also sends out sodium, because sometimes sodium leaks into the neuron, and we don't want this. So the mnemonic that it uses is, if it's K, then it's okay, but if it's Na, then send it away. So there are two forces at play here. We have diffusion, which is driving the potassium out intracellular environment, and we have electrostatic pressure, which is pulling the potassium in. And at the same time, we have this sodium-potassium pump at play, which is trying to keep the potassium concentration at a set level on the interior of the neuron. This set level typically is about negative 65 millivolts. In addition to the potassium channels, which with their selective permeability, and the sodium-potassium pumps, which are actively pumping the sodium out of the neuron and pulling in the potassium, we also have what we're going to call the voltage-gated sodium channels. Voltage-gated sodium channels are channels that are permeable to sodium, but only whenever there's a change in the charge of the interior of the neuron. At resting potential, the inside of the neuron is negative 65 millivolts compared to the exterior of the neuron. However, if we were to raise the charge, which could be done by letting in cations, at some point the charge might reach negative 40 millivolts. That's what we call threshold. The process of going from negative 65 millivolts to negative 40 millivolts is a process that we're going to call depolarization. And when this occurs, we see the beginning of what we call the action potential. The action potential begins in the axon hillock, because the axon hillock is the first place that we have a dense distribution of voltage-gated sodium channels. When the pH, or whenever the electrical charge reaches negative 40 millivolts, the voltage-gated sodium channels open up. When these channels open up, sodium is drawn to the interior of the neuron. You might ask, well, why would it go to the interior of the neuron? There are two reasons. First, diffusion, and the second is electrostatic pressure. The interior of the neuron is negative. Sodium, which is a cation, is positive. The different charges drive the sodium into the intracellular space, as well as the second force, which is the sodium's going down its concentration gradient in a process that we call diffusion. So there's both diffusion and electrostatic pressure at play, which are both driving the sodium into the interior of this neuron, which had a potential of negative 40 millivolts at threshold. Now that we also have sodium inside of the neuron continues to depolarize even further until we reach about 40 millivolts. Now this is positive 40 millivolts. And at this point, if positive 40 millivolts, the voltage-gated sodium channels close, and the voltage-gated potassium channels open. At this point, when the intracellular space reaches 40 millivolts and the voltage-gated potassium pump, potassium is driven out. Well, why would the potassium leave the inside of the neuron and go to the extracellular space? It goes quite rapidly for two reasons. First, the potassium is traveling down its concentration gradient, a process that we're going to call diffusion. And in addition, it's traveling from a positively charged intracellular space to a negatively charged extracellular space. Because again, we have all this sodium and potassium in the interior of the neuron is actually positive at this point. So the potassium is driven by electrostatic pressure out of the neuron. So at this point, we actually end up with the opposite of what we started with. Originally, the interior of the neuron was negative 65 millivolts, and it was saturated with potassium. Now it's saturated with sodium. And the concentration of potassium is higher outside in the extracellular space. At this point, there's a deactivation of the voltage-gated potassium pumps. And we return to one of the original pumps in this process, which is going to be the sodium-potassium pump. And that's going to restore the resting potential because, again, the sodium-potassium pump is going to drag potassium back into the interior of the neuron, while it's dragging sodium to the extracellular space. And once that it's actually done this, the charge will actually return back to the resting potential. But there's actually a brief period where the neuron, the intercellular space of the neuron is actually going to be hyperpolarized, meaning that there's actually a charge even further negative, even more negative than negative 65 millivolts. We would call this time, we would say that it would be negative 75 or negative 80 millivolts. We call this the refractory period. In fact, the refractory period actually begins as soon as the potassium begins to exit the neuron. And this refractory period is a period where the neuron is rendered to inactivity. There is no change. An action potential is not being able to be fired. Then finally, the sodium-potassium pump restores the neuron to its initial state, where the intracellular space is saturated with potassium. And the extracellular space outside of the neuron is saturated with sodium. So we will now say that the neuron has returned to its resting potential. And that is going to be negative 60 millivolts. So we have returned to the initial resting state. And that is how the action potential occurs. So here we have the resting potential at negative 60 millivolts. Again, this can vary. Maybe it can go up to negative 50. Maybe it can go to negative 65. Whenever the charge is actually raised, we call this depolarization. And when it reaches negative 40 millivolts, that's what we call threshold. At that time, the voltage-gated sodium channels open, And as the sodium rushes in, the charge reverses to positive 40 millivolts. At positive 40 millivolts, that activates the voltage-gated potassium channels and deactivates the voltage-gated sodium channels. This drives potassium out of the neuron, which lowers the charge. And at negative 60 millivolts, there's actually a closing of the voltage-gated sodium. At this point, when the charge reaches negative 60 millivolts, there is actually a closing of the voltage-gated potassium channels. And there's what we call a hyperpolarization. As you can see, the charge is below resting potential. At this point, the sodium-potassium pumps go to work, and they transport the sodium out of the neuron while transporting potassium into the neuron, restoring it to what we're going to call resting potential. There's an important period of time, another stage I want to talk about that extends from the point of highest charge to the point of lowest, and we're going to call this the refractory period. The refractory period is the period of time in which the neuron cannot generate another axon potential. And the refractory period ends once the neuron is restored to its resting potential.